Okay, we're going to go over chapter 12 and try and get through this so that I can get it posted on Canvas for you guys. Let me share my screen. This chapter is over genetics and genomics. This is chapter 12. Genetics is the study of genes and their role in inheritance, how a cell operates, proteins it makes so that the protein can correctly control how quickly a cell grows, how often it divides, and how long it lives. It determines how certain traits or conditions are passed from one generation to another. And this all began with a simple pea plant experiment in the 1860s in Austria. And I bet you guys already knew that from your basic studies. So genomics is the study of an individual's genes, a genome, and how they interact with each other and person's environment. This includes the study of complex diseases such as heart disease, asthma, diabetes, and cancer. So here we have a little picture. I bet you already know that there are 23 pairs of chromosomes and that 22 of those chromosomes are homologous. And those are termed autosomes, which are the same in both males and females. The 23rd pair is the sex chromosome, and that's made up of, if you're female, an XX chromosome, and if you're male, an XY chromosome. So here's a picture of a DNA strand. That kind of looks like a ladder that's twisted, and it's composed of 4 different types of proteins: cytosines, thymine, guanine, and adenine. And they specifically pair together. Cytosine always pairs with guanine. Thymine always pairs with adenine. And then the sequencing is different depending on what chromosome you're building or gene you're building. And they're always strung onto the backbone of a sugar phosphate. We also have dominant recessive types of chromosomes. We look at genotype and phenotype. We look at locus positions on the gene and the chromosome. Okay, so this is a picture of representation of transcription and translation. Transcription and translation is a process of protein synthesis. It's a 2-step process. And within this process, mutation can happen. There are 2 types of mutation. Germline mutation is a mutation that is inherited from your parents and that you can give to your kids as you reproduce. An acquired mutation, or also called a somatic mutation, is something that happens during life. It may be a result of an experience of exposure to something, an environmental thing, but you cannot pass that on to your kids. Also, as you age, your DNA ages, and sometimes it just simply mutates and it doesn't fix itself. So that's another style of mutation that can happen. All right, so this slide represents mitosis, and mitosis is a type of cell division that results in the formation of genetically identical daughter cells. Before a cell division, the chromosomes duplicate, and each new cell, each daughter cell, receives an exact replica of the chromosomes from the original cell, the parent cell. Meiosis occurs only in sexual reproductive cells. In meiosis, the number of chromosomes is reduced, resulting in half the usual number of chromosomes. Therefore, an oocyte and a sperm cell contain only a single copy of each chromosome, whereas all the other body cells contain duplicate of each chromosomes. So let's talk about some inheritance patterns. When we talk about autosomal dominant traits, Autosomal dominant are heterozygous. This is a trait that is a single gene pair mutation of a chromosome. It is a dominant prevalence over a normal cell. When we talk about autosomal recessive trait, this is a 2-gene pair mutation. So you may not see this come out in a person because it can be a recessive trait, because there will be one normal gene paired with an abnormal or a mutated gene. But that makes the person a carrier. So if you have 2 parents that are both carriers, they have a chance of giving their carrier gene, each of them giving their carrier gene to an offspring, and that offspring then could not have a normal gene to override the mutated gene, and they would be positive for that mutation. All right, we also look at sex-linked recessive genetic mutations. So X-linked recessive disorders are caused by a mutation on the X chromosome, and this usually only affects men because women who carry the mutated gene on one X chromosome also have another X chromosome to compensate for that mutation. So women who carry the mutated gene can transmit it to their offspring. X-linked dominant disorders do exist, but they're very rare. We have multifactorial inherited conditions that run in families, but these do not show the same inherited characteristics as the single gene mutation conditions. All right, so let's talk about the Human Gene Project. This was a mapping of the entire human genome. This was finished in 2003. People have been doing data analysis, and through that data analysis, the goals have been to improve the diagnosis of diseases, to allow earlier detection of genetic predisposition of diseases, to determine risk assessments for genetic-related diseases, and it also assists in matching organ donors with transplant recipients. This has been a huge undertaking, and it's helped a lot of people. All right, this is in your book. This is a page from your book. This is a family pedigree, and usually family pedigrees show traits over at least 3 generations. This only shows 2 generations, but a true family pedigree is over 3 generations, and it just kind of maps out how disorders fall out. The example A shows an autosomal disorder, B shows an autosomal recessive, A, autosomal dominant, B, autosomal recessive, and then C, X-linked recessive. So take a peek at that. Family pedigrees are important, and you probably need to know about family pedigrees. All right, so let's talk about the classifications of genetic disorders. So we have single gene disorders, and these are things like cystic fibrosis, sickle cell disease, polycystic kidney disease. This is a single gene mutation. It's relatively rare among genetic-related disorders, but if you got it, it's a bad thing. We also have multifactorial gene disorders. That sounds like a mouthful, but really what it is the risk factors for genes make you predisposed for something, and then you combine that with environmental and lifestyle changes. So you might have a gene for heart disease, but when you combine that with bad eating habits, an inactive lifestyle, it's like being in the wrong place at the wrong time. Now, the good news here is that you can control some of this. Small inherited generic variations interact with environmental factors. You can change those. You can do something about it. This last bullet it says it's rarely caused by hereditary mutations in a single gene, but I give you breast cancer. We have isolated that the BRCA1 and BRCA2 gene is a mutation that can cause breast cancer. So we are identifying some of these things. So rare, but yeah, it does happen. All right, let's talk about epigenetics. And this is the environmental influence that can happen on genes. So genetics and environmental factors interact to influence a person's predisposition to different diseases. So this epigenetics is a study of chemical modifications on specific genes, gene-associated proteins of an organism. Environmental factors can alter the way our genes are expressed, meaning which genes are switched on or off. So if you think about some of the things that have happened to some of our service guys, like some of the guys that went to Afghanistan were exposed to Agent Orange. Maybe it was Iraq. We also have Vietnam veterans that were exposed to, maybe that's where Agent Orange was. No, I don't think so. Think about the nuclear bombs that were set off in Japan, the exposure to that. There's been all kinds of environmental things. So that all falls into the category of epigenetics. All right, genetic testing. This is something that is becoming more and more popular. There's a couple of things I need to say about this. First of all, it's difficult to interpret and explain the results. Just because you have a positive result, that doesn't indicate that something bad is going to happen. It just says that a change was found. It was found in a gene, maybe a chromosome, or a protein. It does not establish the absolute risk of developing a disorder, nor does it predict the course of a severity of a condition. It's just an indicator. What it does say is, hey, we need to look at this further. So at that point, you recommend to the patient that they seek genetic counseling, and that's the big deal. They need to go to someone that has been specially trained in genetic research and counseling. Further testing may be required to confirm negative results. Okay. The other thing that's been going on is at-home testing. Things like Ancestry.com, 23andMe, and there's a few more that have come up, have been doing the at-home spit tests. And I have to admit, I'm a member. I think it's pretty cool. My whole family has done 23andMe, and it's fun to see where you come from. And there's been a lot of new regulations that FDA has lightened up on. So they are doing a lot of emails saying, hey, participate in this. You're not, your DNA says that you're not at risk for that, and that's fun. But you have to take it with a grain of salt. So always instruct your patients that before they believe anything, they need to go to a genetic counselor. And if someone, comes in and says, gee, what do I do? Listen to them. Ask them the open-ended questions. How does that make you feel? What do you think? And then refer them to a genetic counselor. This slide talks about pharmacogenomics. And this is just a little bit confusing, but pharmacogenomics is the study of genomic variations associated with drug responses related to multiple genes. This examines the entire genome, not to be confused with pharmacogenetics, which is the study of genetic variability of drug responses related to a single gene variation. So it's kind of the same thing, but the second one is really narrowed down to a single gene variation, if that clarifies it at all. Since this has been singled out in your PowerPoint, I would say this is probably a pretty important point. Again, I'm not the test writer, but this has been singled out, so I would look at this. Let's talk about gene therapy. Gene therapy is the treatment of a specific portion of a gene. We know that there's an error on this gene, so we can go in and treat and fix a specific portion of the gene. Apparently we have that capability. Now you have to realize that just because we fix that gene, that doesn't mean that that fix is going to be passed on to the offspring. It's just fixing it in the person. There is another type of therapy called stem cell therapy, and there's two types, embryonic and adult. There is some discussion going on with both of these. But stem cells are basically unspecified cells in the body that have the ability to remain in an unspecified status until needed, and while they're in that unspecified status, they just keep dividing. Once they're needed, they can differentiate and develop into specific sites. Embryonic cells are a little bit more malleable and can do more than adult cells. But there is controversy over this. So please go to your book and read more about this. But stem cell search shows a lot of promise and has been stopped in a lot of areas because of the controversies. Okay, let's go back to there we go. I went to the wrong mouse. Let's go to genetics. I think this is the last slide. So the fundamental things you need to know are the fundamental knowledge of genetics and genomics. And that's just reading through this chapter and highlighting on some of the things that I've told you. You need to know about HIPAA. And the abbreviation here is wrong. I thought I had that fixed, but it's wrong. It's the Health Insurance Portability and Accountability Act. It's HIPAA. And that guarantees privacy and security for health information. And that is utmost importance. You need to know about GINA, the Genetic Information Non-Discrimination Act. And why is that important? Well, let's consider privacy and non-discrimination. Let's say I go get genetically tested and I come back positive for one of the breast cancer genes. Okay, so I go through genetic counseling and sure enough, it's correct. So I'm probably looking about doing something. I might decide to do a preventative surgery. So if I'm looking at a new job, an employer might look at you and say, I'm not going to hire you because you're going to ding my insurance for a surgery. And you might also end up with cancer, and that's going to ding my insurance too. Well, GINA protects that non-discrimination portion and puts a non-discrimination portion in place. You have to remember to refer to qualified healthcare providers for interpretation of results. And then always do a family health history. That's so very important. Okay, and I think that's the end of the slide set. Yes, that is the end of chapter 12. And I will stop the recording here and get ready for the next chapter.